everyone for tuning in and for letting us into your universities, offices, and homes during this Corona times. I like to start with something funny about this webinar. A friend of mine told me that during this coronavirus shutdown, he has been stuck in his house so long, he's already been through 32 books. I said, wow, that's amazing. He said, yes, it has been a lot of coloring. Say it like you mean it. Yes, it's a celebration, not a formal webinar. It's a gathering of more than 550 scientists from 67 countries. And I hope you enjoyed the sparkles in the beginning. Thank you for being with us on Zoom, as well as watching us live on YouTube. I, Manju M. Gupta, on behalf of International Symbiosis Society, along with my team members, extend hearty welcome on this World Symbiosis Day, which is also International Friendship Day. Before we begin further, I would like to welcome our guest speakers. I would like to formally introduce and welcome our guest speakers. Professor James White, Professor Virginia Weiss, and Professor Mark Andre. And I suppose Mark is having some problem in just getting into this. I will just fix this first and then go ahead.
I worked out which camera I was on.
I don't know why he's not joining. He is around in sending me the mails, but he is not able to join. So I will just quickly introduce everyone to the first to the speakers, and I'm just wondering where is Mark? Just give me 1 minute. He's not here.
He's on WhatsApp asking to get in, so I think he doesn't have the right link.
How do I help him?
I will put the link on WhatsApp, so you don't worry about it.
Yeah, please do that. So I would, yeah, please do that. So starting again, I was just introducing and welcoming my guest speakers for the day. We have Professor James White, the first speaker. He is from Rutgers University, USA. Then we have Professor Virginia Weiss. I think for the audience, I'm starting it now so that audience should know that whom I am referring to. We have Professor Virginia Weiss from Oregon State University. And the third speaker is from Natural History Museum, Professor Mark Andre. He would be with us anytime. He is around, he is in touch on emails and WhatsApp. He has not just shown his face, maybe soon he'll be here. And then apart from this, I like to introduce my executive International Symbiosis Society executive team also, so that, so that everybody knows whom we are referring to. So we have Professor Simon Davey. He's from University of Wellington, New Zealand. He is the president of International Symbiosis Society. Then we have Professor Sharon Doughty. She's here. Sharon is here. Yeah, she is she's from University of Washington, and she is Vice President for Education in our ISS. Then we have another Vice President, Dr. Christina Cruz.
Hello.
She's here for quite a long time. So it is so good to see you, Christina. She is also Vice President in the society. And the third Vice President in the society is myself. And I come from Sri Aurobindo College, University of Delhi, India. So that was it. Then we have Secretary. She is Dr. Silvana Munzi. She is from University of Lisboa, Portugal. And yeah, Mark is here. No, Mark has entered from the login of Virginia probably, because in front of Mark, Virginia is written. Anyway, it goes. So Mark is here. Then who else is? Eric is remaining. Eric is from University of Mississippi, and he is treasurer of our society. right? So I would come back to Mark. Mark is from Natural History Museum. Mark, you can wave for everyone. Everyone was just waiting for you and missing, yeah. And there is light is little bit less on your face, maybe. You can add some light if it is possible. Right, then we have, then I would like to introduce or maybe welcome Dr. David Richardson, Professor David Richardson. He is editor-in-chief of our journal, Symbiosis. And I would also like to extend, he's joined us from Canada. I would also like to extend I would like to acknowledge the Springer Nature for having sponsored this webinar. And they have offered lots of things, whatever was possible to our registrants in this webinar. So that was the general introduction of everyone present here. I hope I have not missed anyone. And then before we actually begin with science, I would just request Professor Chris Walker, I have introduced to everyone, and he happens to be my teacher when I was in UWA with Lynette Abbott, and Chris was also there at that time. So I have learned a lot of mycorrhiza from him, yeah. So then we have, yeah, so I would invite Simon to present his inaugural address for the function. Yeah.
Thank you, Manju. And welcome everybody. I'm going to, excuse me, I'm going to keep this very short because I'd like to get on with the science. But as Manju says, this is the first time that we've held this webinar. And I'm just going to pop up a couple of slides here and just talk over them. But before I do that, I really just want to say welcome to everyone. I can see all the names popping up in the chat box. I'm sure there are lots more people out there. And you know, we're going to be polling a few questions later, but one is whether we think it's worthwhile doing this way more regularly, you know, potentially annually, and we're going to be asking that later. But first of all, so I'm just going to bring up my slides. Can we see this menu?
Yes, yes. Everybody can see that, yes.
Just get rid of that. Hang on. Right. So we've had lots and lots of people engage in this process, and I do want to say thank you to Manju. This was actually Manju's idea originally. She approached me and asked whether she thought this would be a good idea, and it's been a huge success. We've actually, this slide, even though I put this together last night my time, is already out of date. We had about 60 participants from about 60 countries who registered as of last night. Today, this morning, I believe it's 67 different countries. And you'll just see from the map here, from the little circles there, which places have the most participants. So the US, India, Brazil, and a number from Europe. But really what's really struck us is the number of people who have joined us from all far-flung corners of the world. And you know, when we run the Symbiosis Congress, which runs every 3 years, you know, we get people from all over the world, but not this coverage. And I think it's really fantastic that so many people are able to engage. I apologize if your country is missing from here, but this I think this really is you know, wonderful that so many people can join. Now then, I'm just going to click there. And so Manu's already said a little bit of this. I just want to take the next couple of minutes to say a little bit more about the society for those that don't know, or quite honestly, for even those that are members. And you know, the society, you're going to hear lots today about the history of the society, and also some of the key aspects of research that are going on through members of the society. But the ISS or International Symbiosis Society has been really started to arise from the 1980s with the onset of the Symbiosis Journal, which David will talk about later. And then a series of congresses and most recently in Oregon. And Marc Andre is going to be talking about the next meeting, which will be in Lyon. But a number of other meetings, the first one being in Israel, and then it progressed all the way through you know, Canada, it's been in Portugal, and elsewhere. And David is going to talk a little bit about that, and James will talk a little bit about So Manju introduced the current executive committee, but I really just put these up because I wanted to indicate what an international society this is. Even our executive team, which is always a challenge when we're scheduling meetings, cover 4 different continents. And we also have a student rep, Ashley. And Ashley will be talking later about the involvement of students in the society. And that's a relatively new initiative that we've had a student rep, but it's really been instrumental in getting things like our YouTube and Twitter handles and so on set up and active, because of course, as it won't be as any surprise, but the people actually on the committee are not so active in those areas. So having the students involved has been really, really important. So you'll hear about the Spring of Nature Symbiosis Journal later, and that's a very important way in which we disseminate research, and also through the Congress. So is the other way we do that. But I see this webinar as being a potentially really important way that we can reach a wider audience and do it more regularly to celebrate World Symbiosis Day. Now, the society has, its main goals are really to bring together community, which we're doing here, but also to share our research findings. And also we have, as you'll notice, we have 2 vice presidents for education. One of our priorities is to actually encourage the embedding of symbiosis in curricula at high school level and also at university level. And so we try and take a proactive role in doing that. And our memberships typically ranges anywhere between about 350 and 500, depending on where in the conference cycle we are. But of course, we're very keen to increase that as well and get lots of people from different fields of symbiosis represented. And currently, just on the little strip of images on the right-hand slide there, shows some of the areas. So whether it's plant microbial symbiosis, so mycorrhizae and rhizobium, whether it's aphids and other insects and bacterial symbionts, lichens, bioluminescent squid and their bacteria, or myself, I'm a coral reef biologist, and Virginia will be talking about coral reefs later. But of course, there are lots and lots of other types of symbiosis that are also represented through the society, but we're keen to get more. And so we play a very important role in disseminating this information around these symbioses, but also encouraging conversations between individuals who work in these different disciplines. So with that, I'd like to say thank you and welcome to everybody again. And I am going to close down my screen and hand back to Manju. So welcome and thank you.
Thank you, Simon. Time to begin with keynote presentations. Before I invite Sharon to read short profile of Professor James White, I would like to remind all the participants that there is a Q&A icon at the bottom of your screen where you can directly send questions to the panelists. It is different from routine chats and would really help us to address all scientific questions in an organized manner. Yeah, so I would just request Sharon and Professor White to continue with first keynote presentation. And maybe I can put, and everybody else can put their mics and cameras off. Yeah.
Okay, yeah, it's my great pleasure to introduce Dr. James White today as our first keynote speaker. He's a professor of plant biology at Rutgers University. He received his bachelor's and master's degrees, both from Auburn University in botany and plant pathology. And his PhD was in botany mycology from the University of Texas in Austin. He's authored over 230 articles and has been the author and editor of several reference books on endophytes, the microorganisms within plants. He's a fellow of the American Association for the Advancement of Science, associate editor for the journals Symbiosis, Fungal Ecology, Mycoscience, and Scientific Reports. And he also serves as the chief editor for the Plant Microbe Interaction section of the journal Microorganisms. The title of his talk today is Plant Endophytism, Rhizophagy Cycle, and Why the International Symbiosis Society is Important. So thank you for your presentation.
Thank you, Sharon. Okay, I'm going to try to share my screen now, see if I can this to work. Okay. Let's see if I can get it to, it is actually not forwarding. Okay, here we go. Okay, so I'm very, very happy to be here. Very happy to be invited. I think I was invited mostly because of early on, many years ago, back in 1997, I was involved with the helping to found the International Symbiosis Society. And at that time, Margalit Galoun, Israeli scientist, had begun actually the journal Symbiosis, and she started to do some international symbiosis congresses. So there was beginning to be this community nucleating around symbiosis. And along with Margalit Galoun, who was the inspiration, but John Lieb actually became the first president. He was a scientist at CUNY City University in New York, worked on foraminiferans, but he became the president. And Charles Bacon of the USDA became the actually the treasurer of the International Symbiosis Society. And I became the secretary, the first secretary. So I had all the paperwork that I had to do in setting it up. But we founded the society and got it started and passed it on to new groups of people who carried it on. One of the scientists that published in the journal Symbiosis was someone very famous, Lynn Margulis, and her very early endosymbiosis research. And she published in Symbiosis partly because the journal had reviewers that were open to her new ideas. And this is kind of the value of the International Symbiosis Society. It's composed of scientists that are open to thinking about symbiosis and evaluating symbiosis ideas. So I think the International Symbiosis Society continues to have that value, that it's a place that's open for symbiosis research. The research that I'm going to talk about has to do with endophytes, microbial endophytism into plants. And these are microbes that go into plant tissues. They could be fungi, they could be bacteria, they could be, in some cases, algae. But they go into the tissues and they show no symptoms. They're basically asymptomatic. They're not disease organisms. They don't have negative effects. In fact, usually they have positive impacts on plants. And these endophytes basically are in all plants. There's not a plant that doesn't have endophytes in them. In fact, it's not just one endophyte. It actually is an endophytic community in plants. And there may be both bacteria, there may be fungi there, but usually there are plurality of them, and they have different effects in those plants. Back in 2010, and this is the work I'm going to talk about now, has to do with this process called rhizophagy. Rhizophagy, rhizo meaning root, phagy meaning eating. And this is basically a process where back in 2010, it was discovered that plants were internalizing microbes into their roots. And some investigators in Queensland, Australia, this is a young lady here who was actually involved with that research, but there's a whole group there that did this work. And what they found was that plants are internalizing bacteria and yeasts into the root cells. And what they did is to tag these microbes with GFP, and then they tracked them into the plant tissues. And over time, over a period of days, they saw that those microbes, the fluorescence disappeared. And so their idea was that these plants are consuming these microbes and degrading microbes as a source of nutrients. And they published a paper in PLOS One, you can see listed at the bottom here. They titled it "Turning the Table: Plants Consume Microbes as a Source of Nutrients." So that really is the start of this story of rhizophagy. And some of the articles that we published on this, there's several, now actually numerous, but here are three. The first one actually, and this is illustrative, listed here, was actually published in Symbiosis. And of course, that's because Symbiosis is a place where they were open to these new ideas. Later articles published other places you can see here, but the start was really in Symbiosis with this line of work. What we discovered in my lab is that this rhizophagy is actually more of a cycle. And this diagram of a root, you can see it's just kind of a cyclic, saw the red circles here, but microbes in the soil are attracted to the root tip. There's an exudate zone there. And so the plant essentially is cultivating these microbes in exudates at the tip. Some of those microbes are internalized into the root cells, into the epidermal cells of that root, just around that meristematic, that tip meristem. And that's where really the cell walls of the root cells are most plastic. They're not hardened yet. So the microbes go in, we don't exactly know how. There's a couple of ideas, I won't go into them now, but they essentially are internalized. Then they go into the cell, they go into the periplasmic space. The plant then secretes superoxide onto them. And that will actually take the cell walls off of the microbes so they become protoplasts, and they continue to be bombarded with this superoxide. But these microbes are replicated inside the root cells. I didn't mean to do that. Okay, so they're replicated in the root cells, and then they trigger root hair elongation. And as those hairs elongate, the microbes are ejected out of the tips of the hairs. So they go back out into the soil, they form their cell walls, reform their cell walls, and they go out and they acquire nutrients. And then again, they're attracted back, and then they're bombarded with reactive oxygen in the cells, nutrients are extracted and so forth. That's the rhizophagy cycle. Okay, so a little bit about the rhizophagy cycle, some of the details. Okay, so this actually shows a grass root. And this is Phragmites. You can see the microbes around. This is where they're cultivated in the exudate zone. These exudates have sugars, they have amino acids, they've got organic acids. Basically, the plant is feeding these microbes. The plant is attracting these microbes to the root, and then it's internalizing at the epidermis. That brings those microbes into the root cells. And okay, then once they're inside, they actually go into the area we call the periplasmic space. That's the area just underneath the cell wall, but outside the plasma membrane of the root cell. So they're kind of trapped in there underneath the cell wall. Okay, initially they will have their cell walls, and we can look in there with microscopically, and we can see the bacteria inside the cells, inside the root cells. But after they're hit with reactive oxygen, superoxide, their actually walls are lost, and they become these spherical protoplasts. The image to the right shows these protoplast stages. And these protoplast stages replicate very rapidly. So the plant can remove the wall and then can replicate these, essentially clone these microbes very rapidly, make a lot of copies, make a lot of clones of those microbes. And that's what happens once they're inside. But at the same time, nutrients are being extracted from these microbes. At least that's our concept. And we can measure nutrients being lost from the being acquired by the plant. Okay, this actually is a picture of a root cell, and you can see the little brown structures in there. Those are the protoplasts of the microbes, and they are in that. They're in the root cells, and they're being replicated. You can see they're making these little chains. That's not how bacteria normally divide. Normally they'll divide when they have the cell walls. So these are without cell walls. Okay, so this is a diagram. This is a diagram that shows a plant cell and a root cell. And you can see the cell wall out here. You see the membrane in here, and you see the microbes in there in that periplasmic space. So that's just outside that membrane. And the enzyme that produces the superoxide is an enzyme we are sometimes called NOx enzymes, or NADPH oxidase enzymes, and that is those are actually in the membrane of the root cell, and that enzyme will take molecular oxygen from the air and will turn that into superoxide. And superoxide is highly potent, so it can hit those microbes and oxidize the cell walls off of those microbes, and also can also make those protoplasts leak. But it also break down some of those protoplasts. So some of those protoplasts are fully degraded. Okay, this is one of our experimental plants. This is a sedge from the desert or the island of Bonaire in the Dutch Antilles, and you can see it's growing on these limestone formations. This sedge will grow pretty much without soil, but what they do have a lot of is microbes. And this actually shows that. The image to the bottom shows a root hair of that sedge, and the red areas, those are where the bacteria are. You can see the arrows there indicating that those are bacteria in the periplasmic space. This is not static. Okay, this actually is moving. In the image to the top, you can see this is a moving image. It's pretty much how fast it's going. And those bacteria are being rotated around those hairs like that. So they're in constant rotation. And that's important because that enables the breakdown of gradients, that are nutrient gradients that occur between the plant root and the microbe. And so there's a maximum flow of nutrients between the plant and the microbe. Okay, this is a diagram that actually shows how what happens is the microbes will be moved to the tip of the hair and then they'll be ejected. And we call this actually cyclosis expansion wave mechanism for microbe expulsion from root hairs. And what happens is you see the arrows here, that's the cyclosis or movement of microbes around. So they're moved to the tip and they accumulate at the tip. And some of these tips will get really fat with microbes. All these little black dots are the microbes. And then what happens at the base, there is an expansion wave that starts at the base of the hair and expands all the way to the tip. And then it pushes those microbes out of pores at the tip. So they go back out into the soil. And once they're back in the soil, exudates will go through the hair and then will feed those microbes outside of the hair, and they'll reform their cell walls and they'll go back into the soil and acquire nutrients. Okay, so the way this expansion wave happens is we think it's actually something called potassium loading. And that is that potassium is pumped into the vacuole. There's a vacuole in this hair. It goes into the vacuole and then water rushes in. And when that happens, it makes this expansion wave that goes from the base and propagates to the tip. This actually shows tomato, and you can see this expansion wave happening. If you look at 3, you'll see the very where the tip is. And if you look right at that tip there, you can see there's 3 or 4 pores right at the tip, and you see those microbes going out. You can see that expansion wave starts at the bottom and goes all the way to the top and pushes out those microbes. Then they'll reform their walls once outside. Okay, this is a nutrient absorption function. We've measured nutrients here, and other people have measured nutrients in the rhizophagy cycle. Among the nutrients is nitrogen, for example. I had one visiting scientist that did some work with winter wheat. And what he did was to remove the microbes off winter wheat and then put them back on one at a time. And we do this with seedlings. We can remove the microbes by taking seeds and sterilizing the seeds, get the microbes off, and then put them back on, isolate them and then put them back on. And then he measured, cut the shoot material and then measured nutrients with and without the microbes. And this shows nitrogen result, and here without bacteria compared to with bacteria. You can see it looks like about a 30% increase when those bacteria, the bacteria in this case happen to be bacillus, different species or different strains of bacillus. For phosphorus, very similar result. See without bacteria here, but then with the bacteria, you see it looks like probably about 20%, 25% more with the microbes there. Potassium, you can see one of these microbes was extremely good. Looks like 3 times maybe the amount of potassium that you have in this no bacterial control. The other's not quite as good. The same thing, you see a similar thing with iron and zinc, manganese. So other nutrients are also, we're still trying to figure out, exactly how the nutrients are being transferred. In some cases, we think nutrients go from basically from cell walls. There's a lot of protein in cell walls. In other cases, we think, for example, with potassium, we think that leaks out of cytoplasm. Okay, so this work, that's just a summary of this rhizophagy cycle, the work, these people who were involved over the years, and they all contributed little bits. I guess I'm out of time. Okay, thank you very much.
Yeah, thank you, James, for your such a symbolic presentation, which was a perfect amalgam of information about the symbiotic society and the endosymbionts. After seeing pics of so many students around the globe in your group, I just wanted to share this knowledge of mine that Professor James has been quite generous in taking international students both in PhD and postdoc, as I came to know in the Lisbon conference. So now the time for question answer segments, and I could see thank you everyone for writing the question, and I can understand we get to listen to Professor White on fewer occasions, and there have been many questions, but I would just take one at this stage, and rest of them I would hand over to Professor White. Maybe he would like to answer them live on during the open discussion session, or maybe he'll be answering them here itself. So one question which I selected out of this, which I thought is a simpler one, is maybe Linda Blackwell from, I don't know which place she is from. She has asked, how did you capture the moving bacteria in root hair?
Yeah,
James, would you like, yeah?
Yes. A lot of our work is done in assays in agarose, in petri dishes, and so what we can do there is basically grow our seedlings, because mostly we're using seedlings, with their microbes in these agarose plastic petri dishes, and then we just reverse those dishes, and then photograph through the reverse of the petri dish, and we can get pretty good resolution. We can't go to oil immersion because these petri dishes are thick, but we can get high dry and get some good little films of the microbes inside the roots. They actually are going quite fast, so those are the speeded up just a little bit, but they're like 10 to 11 micrometers per second. So we really have quite fast movement inside these root hairs. The cyclosis happens all the time. It's not at all a static process. It's moving. So there's a lot of activity there.
Yeah. I hope she is happy and satisfied with this answer. And maybe if she wants to take this up again, she can take up an open discussion again. And I think before we move to the next keynote speaker, I just want to run a quick poll for one question about the society. So this is for the audience to answer. Please do attempt this. And I have just launched this poll, and this would really help us in improving ourselves. So I would just request and wait for the polling to be over. I think it is going to take 1 or 2 minutes. So I would request everyone to participate in this. Yeah, so 70%, 78% of the participants, they voted, And of the 78%, 99% said that we should go for it and they would like it to be made a annual event, and 0% said no to this, and 3 out of the total, maybe 1% were undecided. Yeah, so I can just share the result of the polling. Yeah, this is the result of the polling for everyone to see maybe. So thank you very much for your overwhelming response to our call of this webinar. And thank you again for supporting us all the way. So going further with this, I would invite the next keynote speaker. And I would just request Simon Simon and Professor Virginia, I would just request Simon to introduce the academic profile of Professor Virginia. And maybe because both of you are from the same field, you can selectively select the questions which can be asked to her. So over to you, Simon. I would just.
Thank you, Manja. I'm delighted to welcome Dr. Virginia Weiss. And Virginia is Distinguished Professor at Oregon State University. And she also, until very recently, and she's probably quite relieved about this, it was the head of department there. She just finished, why she's so excited. And Virginia, just a little bit of background, Virginia completed her PhD in 1990 with Len Muscatine. For those in the coral field, is basically the most famous person in the field. And also then completed postdocs with Margaret McFall-Nye on squid symbiosis and Paul Levine at the Hopkins Marine Lab. She's been at OSU, Oregon State University, since 1996. And Virginia has a very strong track record in the field of the understanding of the onset and maintenance, immune responses, and so on in the cnidarian dinoflagellate symbiosis. And she's also been heavily involved in the development of the Ictasia model system, which I've collaborated with her on quite a bit. And Virginia, in recent times, has become much more of a strong advocate for the conservation of reefs and the application of novel technologies to the conservation of reefs. So with that, Virginia, I will hand over to you.
Thank you so much, Simon. And Manju, thank you so much for suggesting this amazing idea of a World Symbiosis Day. There are almost no silver linings in the pandemic, but I would say one is that we're thinking in science and in academia about really new creative ways to be more inclusive and international. And I don't think conferences will ever be the same. So I think this is an absolutely fantastic opportunity. The next time we do it, I think Americans should have to get up at midnight. So the next time it should be some other time zone that gets the good sweet spot. So with that, I will go ahead and share my screen, and I will get my laser pointer going here. Okay. So 15 minutes isn't long, so I'm going to breeze through just a sort of general background about corals and coral reefs, and then take you through a little bit of what my lab has done all these years, and finish with some of the new recent very serious problems that corals are facing and how we're trying to help with that problem in the field. So as we know, coral reefs are hotbeds of productivity and biodiversity on the planet. They are true oases in a tropical desert where the seas themselves are very, very nutrient poor, and then these reefs are incredibly rich. And they have a huge biodiversity punch. They cover a very tiny amount of the total marine environment, less than 1%, but they harbor 25% of all marine species. And they have enormous ecosystem services impacts to humans. $11 trillion estimated annually to come from the services of reefs. And about 500 million people rely directly on those services. And most of those people are from developing nations with a little voice in some of the very serious impacts that are happening to reefs. So it's really inherent. It's important for those of us in privileged countries to really focus on helping people in these nations that are so, where reefs are so important, to try to preserve them. I'm not giving anybody any new information here. Global warming heads the list of threats to corals. There are many threats, but they all act synergistically with the big elephant, which is global warming. And whereas I used to have to spend time in presentations convincing people of this, now the headlines in popular media and all across everything is about the threats to reefs in so many ways. So for this talk, I'm going to talk really in two parts. One on discovery, that is the regulation of coral algal symbiosis and the role of innate immunity in symbiosis, which is what my lab group studies. And then racing to develop solutions to coral reef decline. And I won't have time to talk about both of these things. These are the two things that I'm focused on: building a global model systems community and working on coral cryopreservation germplasm repositories. So let's first talk about discovery by introducing you to corals. Corals are clonal cnidarians. And so they display this incredible diversity, both morphological, phylogenetic, and functional diversity. So they each on a coral reef, the corals will have different functions that in total drive that ecosystem. When we look up close, we see that in most cases, corals are made of colonies of genetically identical polyps. So this is a close-up of a polyp. And in total, these reefs and their colonies are both bioengineers. So they form the structural foundation of the reef because they calcify, they put down massive calcium carbonate skeletons, but they also form the trophic foundation of reefs. And this is where symbiosis comes in. So they engage in a symbiosis with photosynthetic algae. If we look up close at a tentacle of a polyp, a coral polyp, we can see these thousands of coccoid cells that are deeply embedded inside of the host tissue, inside of host cells. And the central dogma of the symbiosis surrounds nutritional exchange, whereby the algae are in a very high light, safe environment where they are getting inorganic carbon and nitrogen and phosphate from the host. And in turn, they're fixing huge amounts of carbon and passing those back to the host cell. And the symbiosis is so intimate that the algae are intracellular symbionts in these symbiosomes. And this is just an image of dissociated host cells. So this is one host cell. There's the little nucleus right there. And that host cell has probably 4 algae in it. So the algae are taking up almost the entire volume of these cells that are harboring them. The diversity of the symbiosis is not just in the host. There's also the algae themselves are very diverse as well, phylogenetically, morphologically, and functionally. Recently, the group that engage in symbiosis with corals and other things has been replaced into a family, Symbiodiniaceae. Diverse sets of genera. The genera keep coming. I think we're up to 9 or 10 genera now that are represented in this big family. And the key thing to understand here is that host symbiont specificity is diverse, complex, and dynamic. So it's not a one-to-one thing between hosts and symbionts. There's complexity in that, and there's a huge amount of effort and research devoted to understanding that complexity. And in the context of conservation and restoration, we can ask, can specificity be altered to breed or engineer resilient corals? And I'll just leave that hanging. That's something that lots of people are focused on, but not something I'm going to talk about today. My group has studied symbiosis regulation for 24 years. We've been interested in the symbiosis when in health. So when the partnership is engaged in a healthy interpartner communication, it's avoiding immune destruction. So the algae are resident within host cells, but recognized as friend, not foe. So you might imagine that as a teeter-totter where the host is balancing the ability to resist negative invaders, but also tolerate positive mutualists like their symbionts. And this is a dynamic homeostasis that involves a lot of communication between partners. But my group has also been very interested in the mechanisms that drive the breakdown of symbiosis in corals. So, and that breakdown is really about a communication breakdown. It's about stress on the holobiont. And in that case, the corals lose their algae and we move from symbiosis to dysbiosis. And so these are the phenomena that we see on these mass bleaching events where the symbiosis breaks down and you're left with a naked host that has no brownish algae in it. Initial contact between the partners is in most cases something that happens anew with each host generation. And so just to give you a sense, this was work that was done years ago on corals in my group, where most corals spawn eggs and sperm at a certain time of year. They have external fertilization. The larva develops. Larvae then can feed, and in the lab, you can add algae to that food. They drop down to the bottom of the dish, spew out a big mucus string, and trap algae in that string and become colonized. And you can even see this with SEM. These are little planula larvae. Here's a mucus thread extending during feeding, and you can even see the alga that's trapped on that thread, and then ultimately goes sort of down the toilet into the gastric cavity of the host. And so this initial contact is similar to that in other phagocytes. So host cells are nutritive phagocytes that extend pseudopods up and surround algae and bring them into their cells. It's absolutely homologous in mechanism to other professional phagocytes, for example, in our immune systems. And this is just shown here with the macrophage engulfing yeast. And ultimately then these symbionts take up residence in the gastrodermis and they persist. They are not lost. And so one can ask, what are the processes that lead to engulfment and persistence in the symbiosis? So then I'm going to turn to a series of cartoons. It's basically a data-free talk, but it's going to summarize the kinds of things that my group has done through the years. Phagocytosis of microbes involves innate immunity that is very highly conserved from amoeba all the way up to humans. And it typically involves a microbe that has a variety of microbe-associated molecular patterns that decorate its surface that it can't necessarily hide. These patterns are recognized by receptors on a host cell called pattern recognition receptors that in a lock and key mechanism, when engaged, can launch a series of host signaling cascades in an animal cell if it's a negative invader that can kill that symbiont or that negative invader in all sorts of ways, be it apoptosis, launching an inflammatory response, etc. In the case of corals, the Symbiodiniaceae also have these MAMPs, and you can see that these processes are complex. There are many highly conserved signaling cascades in host cells, MAMPs that are on the outside of symbionts, and PRRs that are programmed to recognize those MAMPs. And this information has been acquired through a variety of functional genomic exploratory discovery-based techniques that have been used through the years that we've now identified a lot of these pathways. So what we think is happening, what we hypothesize is happening, is that the symbionts are taken in, and somehow, in ways that we don't fully understand, these pathways to destroy the symbionts are blocked, or that host cell becomes quiescent, and the symbiont persists. And this is really where my group has spent most of its time all of these years. So each of these pathways or genes is, if there's a check mark, it's something the Weiss Lab has worked on, and then the names of the folks who published papers and done theses and interacted with other colleagues in these different in these different systems. And although we don't have a full jigsaw puzzle of information assembled, we have partial bits of that puzzle that continually get reinforced in any new functional genomic study that comes up, where we really are getting a strong picture of what's driving symbiosis establishment and what's also causing bleaching. So if we switch over to talking about dysbiosis, to review then in a healthy partnership, it's apparent that these negative invader pathways are quiescent. They're blocked in some way during a healthy symbiosis. And really the going hypothesis in the field is that dysbiosis and bleaching causes a re-engagement of these immune mechanisms that drives the host cell to eliminate the symbiont, or, and thinking of it another way, the symbiont is creating these pathways or activating these pathways to get the heck out of the host cell. So coral bleaching, we know a fair amount about it at this point. We know that heat stress is the primary cause of bleaching. Heat stress on the algal symbionts destabilizes the photosynthetic apparatus, which in turn causes oxidative stress in the algae. These ROFs can leak into the host, which of course are toxic, but they're also signaling. Heat stress on host tissues also happens, for example, in mitochondrial membranes. Similarly, toxic molecules can be produced from that. There is increasing evidence now that there's a metabolic imbalance caused by the cessation of the symbiont producing photosynthate, which is also a strong player in what drives instability. And at the end of the day, the algae exit the host by one or more of these mechanisms, be it the symbiont degrading in situ, the host booting out the symbiont via exocytosis, a whole host cell detaching and swimming away, host cell apoptosis, host cell necrosis, and I don't even show host cell autophagy here. So there are there's a lot of evidence that all of these things in various ways are at play. We know that corals are differentially susceptible to bleaching. Not all corals bleach in the same way at the same time. And again, one big question in the field is, can breeding or assisted evolution drive coral tolerance and or coral resistance that could preserve reefs? There's an enormous amount of interest in understanding whether or not this is possible, whether, if as human beings we should be assisting in this, and what's more, how fast could it be done? So briefly, I'm going to talk about racing to develop solutions to coral reef decline. And I'm going to talk briefly about model systems. Just to go back to this slide, again, global warming is heading the list of threats to corals. And we as coral biologists are really racing to try to address the decline that's happening to these biomes. And we need to develop solutions for helping corals to survive climate change. There are many possible solutions. Obviously, as single individual scientists, we don't have the ability to lower CO2 ourselves. There are lots of other things that we can be doing to help address the crisis as political, economic, and societal things are happening as well. My areas of effort are in developing Aptasia as a model system for the study of coral symbiosis, because it accelerates these other studies in corals themselves on acclimatization and breeding, on assisted evolution and genetic engineering. I'm also very involved in building global germplasm repositories, but I'm not going to talk about that today. I could talk about it offline or later on. We know that corals are lousy model organisms, right? They live forever, they spawn once a year, they grow slowly, they're huge, they're endangered. They're terrible model organisms. And so if you need you need a playground to really discover things. And corals are not that playground because they are so non-model worthy. So my group and now many others are studying a symbiotic sea anemone, Aptasia pallida, which I call just by a common name, Aptasia. It grows very well in the lab, it's clonal. You can bleach it experimentally. We now have good genomic resources for it. It has a variety of symbiont types, all globally, all of which now have completed genomes or transcriptomes, and all of which are culturable. Many of the symbionts that live in corals are not culturable. They're too obligate, and as yet we don't have them in culture. I've been working with colleagues, collaborators on developing Aptasia as an international network of investigators. At the last ISS Congress, which we had in Oregon, we had a workshop afterwards on Aptasia. We had 54 participants from around the world. We've launched protocols.io site. We have launched a web portal that provides an entree into the resources available for Aptasia, like the genomes for the symbionts, people working in the field, educational resources. We've been promoting Aptasia pretty significantly, and we just were on the cover of Lab Animal in early 2020. So I really strongly believe that these are important ways to engage, to help to try to help coral biologists develop some of the cutting edge genetic, genomic, and functional genomic solutions to help saving corals. And it's really not about me, it's about my many lab groups through the years. This is just a quick slide to show all of these wonderful trainees, many of whom are still in coral biology, but also in other fields like science communication and lots of other things. So my final message is we can't give up on reefs. We have to help with solutions to the reef crisis. We have to focus on the future. We have to stop mourning the past, moving forward, focusing on the future. We have to work together in a mission-driven way, and we have to be cooperative, and we have to emphasize rapid action. And of course, we're always looking for people who are interested in joining this effort to save reefs. And as just one quotation, not even the wise know all ends. So Gandalf was wise himself, and that's my final message. We can't give up on reefs. So I'll stop there.
Okay, many thanks, Virginia. And I am just, we have a lot of questions here as well. I'm just going to pick out one, and this is from, I'm just gonna pick out the final one that came in from Lorraine Ling, who refers to recent work on assisted evolution strategies, and just wondering what your thoughts are on assisted evolution strategies. Only 3 out of the 10 of evolved Symbiosis strains in a recent paper led to resistance to heat-induced bleaching in the host. I'm just wondering what your current views are. I think acknowledging that they might change over time.
Yeah, so these are very hard, very controversial discussions that we need to have as scientists. Is what is the role of humans in saving corals? Should we, we have so many cautionary tales from lots of other systems about biocontrol where we have failed to understand the implications of changing things. My opinion is we don't have enough time to, it's kind of like the metaphor of the COVID-19 vaccine. Like, we don't have enough time to just see if the vaccine works. We need to produce it, and then hopefully it works and we can rapidly push it out. We don't have enough time to discuss the ethics of these things and then launch these efforts. My strong feeling is we need to see if we can, if these ideas of assisted evolution are possible, and then really, if they are possible, if they are not possible, are they? Then we're done. If they are possible, are they scalable? If they are possible, how do they fit into the large array of other solutions that are being developed? And so I think it's one of the arrows in our quiver that we need to investigate. So and not everybody agrees with me, but I think it's very important that we have all options on the table because it's our best chance. The one other thing I will say is that reefs, there are all sorts of reefs, and there are local reefs that are missing, that fishermen need or hotels need, to very locally within an acre try to reconstruct to make their lives better, right? As opposed to, say, the Great Barrier Reef, which has an enormous biome level set of implications. And so those local versus, more global questions really drive the way in which these different solutions might be valuable. So I'll stop there.
Okay, thank you. And I say there are a number of other excellent questions there under the Q&A. So Virginia will either answer those directly or we'll come back to those later in the discussion. And with that, I'm going to hand back over to Manju.
Yeah, thank you, Virginia and Simon. I think without these marine ecosystems, things would have been incomplete. Thank you, Virginia, for covering that. Now I would invite Dr. Silvana and Professor Marc Andre to start his presentation on mycorrhiza. Silvana, I think first you could tell the brief introduction about Professor Marc, and then you will start with his presentation.
Sure, it's a really pleasure for me to introduce you Marc-André Selot. He's a professor at the Musée National d'Histoire Naturelle in Paris and at the University of Koning in China and in Poland. His research focuses on the ecology and evolution of mycorrhizas, but he also has a general interest for symbiosis and its evolution. He is head of the French Botanical Society, member of the French Academy of Agriculture, editor of several international scientific journals, and author of more than 170 scientific papers and 160 published papers. Marc-André, thank you. The ground is yours.
Thank you, Silvana, for this introduction, and also thank you to Manju Gupta for organizing all the facilities there. I'm trying to share my screen with you. Okay, I think you get it. Here we are. So yes, I would like to share with you some general ideas and introduction to the origin and evolution of the mycorrhizal symbiosis, both general topics and some of our latest research. Now, first, I would like to introduce this symbiosis because this model may not be obvious to everyone. The idea is that the roots of plants are associated to fungi, as you can see on this picture, and the mycorrhiza is this dual organ which is made of a soil fungus on the root of a plant. This specific one is a mycorrhiza where the fungus is making a sheet around the root, and this is what we call an ectomycorrhiza. This type of mycorrhiza is found in 2% of plants and is involving ascomycetes and basidiomycetes from the soil, but it's not the main type. The main type is much more difficult to observe directly. You may find it on maize, for example, where the roots look without fungus. And if you look carefully, you can, in the roots, by coloration, see that the fungus is present. A fungus is present which is making hyphae, arbuscules, there's very divided hyphae within the host cells, and vesicles. So this is much more intimate, but it's also a mycorrhiza, which is called an arbuscular mycorrhiza. And this is, for most plants, the case we observe. And the fungus is a glomeromycete, a totally different group of the previous ones. The symbiosis is basically a nutrient symbiosis, a nutrient exchange with sugar from the plant against water and minerals that the fungus has been collecting in the soil. But there is also a reciprocal protection of the two partners against soil pathogens and abiotic stresses. Now, if you look at the phylogenetic distribution of the ectomycorrhizal symbiosis, you see that it's been arising in several different orders of plants. So it's a kind of convergence in evolution of plants. While if you look at the distribution of arbuscular mycorrhizal symbiosis, you can see that it's present in all orders of plant. And this reflects a different evolutionary origin. Let's look closer at the origin of the arbuscular mycorrhizal symbiosis. I would like to show you a piece of stone which is 400 million years old from the Rhynie Chert in Scotland. And you can see that in the rock there are rhizomes of plant which are embedded. And those rhizomes are very well preserved so that you can do some microscopy on thin slices of the rock. You can see that there is a kind of blackish zone. And if you look carefully with the microscope on this blackish zone, you can see that 400 million years ago, there were already arbuscules, hyphae and vesicles in the underground parts of plants. So I think this is the fossil evidence that the reason why the arbuscular microbial symbiosis is so much widespread is that it's just ancestral. Now, there is a diversification because there are now many other types of mycorrhizal associations. We start with this arbuscular mycorrhizal symbiosis, which is ancestral, which was probably arisen as early as the first plant plants. And the question is, what happened next? Well, first, you see that at some point they shifted to non-mycorrhizal. But other groups of plants like Ericaceae, orchids, or many plants with the ectomycorrhizal symbiosis, they shifted to a different kind of mycorrhiza, which means not the same shape, not the same partners. Let's focus a bit on the ectomycorrhizal symbiosis, which is a bit of a paradox. Because if you look once again at the distribution of the plants, which are ectomycorrhizal, you can see that they show that the association has been gained more than 30 times. If you look at the phylogenetic tree of the fungus, you can see that the ectomycorrhizal taxa, the ectomycorrhizal fungi, they arose many times. We know now that they arose more than 80 times in the evolution of fungi. So there were many jumps from arbuscular mycorrhizal to ectomycorrhizal. How was that possible? I would like to make a tour in our exploration of an ectomycorrhizal fungus, which gives, I think, some clues, which is the truffle. The black truffle, you know, is a delicacy, it's something very prized in gastronomy. But for us, it's mainly an ectomycorrhizal fungus. It may be less romantic, of course, but that's an ectomycorrhizal fungus. And this ectomycorrhizal fungus has a specific feature that was very intriguing for us. Where it grows, the arbuscular mycorrhizal plant growing around are very weakly, poorly growing. That is, you have the ectomycorrhizal tree, the truffle are mycorrhizal on the tree roots. And where those ectomycorrhizae are found, you can see that the arbuscular mycorrhizal plants that are growing around, they have a very reduced growth. And because of this reduced growth, we just wondered whether there was not some kind of interaction between truffle and those arbuscular mycorrhizal plants. So we selected some plants from those places, which are called brûlés, because they look like burnt. Brûlé is the French for burnt. We harvested those plants, we looked at very healthy roots, and we tried to detect by molecular tools if tuber melanosporum DNA was present. We found that on 44% of the replicates we investigated, there was a trace of truffle. And if you pile all that up together, you find that 100% of the species tested, they show at least one root with truffle DNA on it. We then tried to see the whole fungal diversity on those roots by using NGS barcoding. And you can see that we found in the roots of those arbuscular mycorrhizal plants from the brûlé, a lot of fungi, which are expected endophytes, pathogens, saprobes, but also a lot of tuber. And not only we found a lot of tuber, but when we were taking similar plants out of the brûlé, away from place where truffle are growing, we didn't find any truffle on their roots. So we had clearly a presence of truffle DNA in this case and not a contamination. We then were able to perform some fluorescent in situ hybridization in the roots of those plants. So you remember, they are arbuscular mycorrhizal plants. And what we see is that we are able to detect hyphae, which are stained by the probes that are specific to truffles. So in healthy medicago roots or healthy geranium roots and many others, we were able to show that in those healthy roots, we did have a presence of truffle hyphae. So what we see is that those hyphae are probably alive because we have a ribosomal staining with those fish techniques. They are colonizing, lining the cells between living cells, so it looks like a biotrophic interaction. And this kind of very loose colonization is, of course, not a mycorrhiza. There is no differentiation. It is something we often call endophytism. So we have an endophytic behavior of this ectomycorrhizal tuber melanosporum species in arbuscular mycorrhizal plants. But it's not the whole story, because if you look carefully at those results, we also found in those arbuscular mycorrhizal roots the evidence for DNA of other ectomycorrhizal species, which should not be there. If you zoom a bit on those ones, you can see that species of Stenophoracea, Scleroderma, Pyronemataceae are present in those roots where they should not be. So the question is whether other ectomycorrhizal fungal species may also be root endophytic. We investigated that by looking at arbuscular mycorrhizal plants in forests where we also have some ectomycorrhizal trees around Paris, simply around Paris. And what we found from 3 sites is that with 50 plants per site, we found a huge diversity of ectomycorrhizal taxa, such as Russulaceae, Inocibaceae, Tuberaceae, etc. We found them in the roots of arbuscular mycorrhizal plants. So now we need to know more what they are doing there, but clearly it seems that ectomycorrhizal fungi are detectable in non-ectomycorrhizal plants. They probably are endophytic, but we need to first check by fish that they are really there, and later also to see what is their effect on the host in this case. But this is opening the door to an evolutionary interpretation of why ectomycorrhizal symbiosis emerge in evolution so often and so easily. Look into the following data. We know it's the conventional idea that ectomycorrhizal fungi evolved from saprobic ancestors. But the question is, how did saprobic ancestors convert into ectomycorrhizal trees? And the suggestion I want to do to you is that they first became endophytic fungi, endophytic partners, and then they turned into ectomycorrhizal. I call this the waiting room hypothesis, where endophytism is a kind of loose interaction, which is a waiting room for evolving tighter interactions, such as a mycorrhizal symbiosis. So the idea is that the transition would be an endophytic colonization. And some of the ectomycorrhizal fungi currently retain the ability to be endophytic in many plants, which is their ancestral state. We, and I will not go into detail into that, were able with Michael Weiss and others to fairly well demonstrate this for a group of ectomycorrhizal fungi, which are also well known as endophytes, the Sebacinales. But I would like to tell you that this story is not only a story that is relevant for the origin of ectomycorrhiza, but also for the 2 other types of mycorrhiza that evolved secondarily from arbuscular mycorrhizal ancestors. In Ericaceae, there are ericoid mycorrhiza, which are involving Heliochiales and Sebacinales, which are well known to harbor a lot of endophytic species. In orchid mycorrhizas, there are 3 taxa, including again Sebacinales, which are known also to have endophytic behavior. So the waiting room hypothesis may also account for the origin of ericoid mycorrhiza and orchid mycorrhiza, but mainly for the multiple origins of ectomycorrhizal symbiosis. So the recruitment of endophytic fungi may have allowed the shift from arbuscular mycorrhizal to ecto, ericoid, or orchid mycorrhiza. I would like to finish now by showing you another trend in the evolution of the mycorrhizal symbiosis, which is a trend to the loss of mutualism. This trend here is not a trend in terms of changing partner, it's rather a trend in terms of changing the functioning of the association. It's a kind of shifting the mutualistic interaction that we were describing for the general functioning of mycorrhizae to cheating. This cheating is observed in plants which are not green, so they cannot reward the fungus with sugar or vitamins or any product that they produce because they are not photosynthetic. Those non-green plants are fully receiving minerals, water, but also sugar from their fungus, and they evolved in many taxa, which are listed here. Some are among Jansians, some are among Ericaceae, some are among Polygonaceae, others among orchids. And I would like to share a first example of that with Neocia nidusavis, a non-green orchid from Europe. You see the root ball, which is the group of those mycorrhiza. And by molecular tools, I was able long ago to show that they were associated with Sebacinales, and that exactly the same species were present on the nearby tree roots where they were performing ectomycorrhiza. So here you have a fungus, which is on the one hand, an orchid mycorrhizal fungus, on the other hand, forming ectomycorrhizae, but it's a shared fungus that allows a carbon flow from the green trees it is mycorrhizal on to mycoheterotrophic plants. In a way, mycoheterotrophy, this heterotrophy based on fungi, is a way of exploiting mycorrhizal networks linking the non-green plant, a cheater in a way, to the green ones and allowing a pipeline for photosynthesis. This nutrition has a very interesting isotopic signature because if you look at the carbon-13 and nitrogen-15 content spontaneous without any labeling of those plants, you can see that the mycoheterotrophic plant are much enriched in carbon-13 and nitrogen-15, which may reflect the spontaneous enrichment of the fungi those plants are feeding on. So that's a very interesting property, that there is a kind of signature, fingerprint of mycoheterotrophy, which is high N15 carbon-13 content. And we went with this tool to investigate the following question. Very often, mycoheterotrophic plants, they arose many times in the same group of green plants. And our idea was, those green plants, are they not already a bit cheater? But the fact is that if you look, for example, of a tribe of orchids, when you look at the fungal associate of those orchids, they also share fungi with the surrounding trees. So there's a potential for a network. Now let's look at isotopic content that may be the evidence for a carbon flow from the fungus, adding up to their photosynthesis. I come back to the same spot where we had this view of a spontaneous nitrogen-15 and carbon-13 enrichment in mycoheterotrophic plants. And now I show you a couple of those orchids which are phylogenetically close to mycoheterotrophic ones. And you can see that their nitrogen-15 and carbon-13 content is intermediate between the green autotrophic and the mycoheterotrophic plant, suggesting that they are mixing mycoheterotrophic and photosynthetic nutrition, resulting in an intermediate isotopic content. In mixotrophic species, we have many other, but I will make it short, we have many other evidence for heterotrophy or partial heterotrophy, because we know also, and we studied this a lot, from reproductive traits to transcriptomic and metabolomic, we know that we have variants, mutants, which are achlorophilus and survive. They survive over years, definitively showing that in those green plants, photosynthesis is not the sole nutrition source. This is the mixotrophy. That is a kind of nutrition that is mixing a bit of photosynthesis and a bit of mycoheterotrophy. It means that cheating may be more hidden because some green plant may be cheater on the fungus, but also it shows that mixotrophy may be a predisposition to evolving mycoheterotrophy by losing the photosynthetic source of nutrients. Now, the main body of cheating mycoheterotrophic plants are based on arbuscular mycorrhizal fungi. And when you go to the tropical forest, you have a lot of gentians or Burmaniaceae that are fully non-green and harvesting carbon from arbuscular mycorrhizal fungi. Recently, we made with a student of the lab, Benoit Perez, a big meta-analysis piling up all the data of what plant is associated to what glomeromycete, adding on the side the phylogeny of the partners to have a bit of a look at what is the specificity in terms of interaction of those mycoheterotrophic plants which are associating with glomeromycetes, so that are cheating on the arbuscular mycorrhizal symbiosis. And in this online published paper, we show that mycoheterotrophs evolve repeatedly. Not a great news, we knew it. That they are more specialized, they have less partners, they are more specific than autotrophs. But they all, and that's very interesting, tend to associate with similar fungi. So not any fungus can be cheated. The oldest emergence is very ancient, and it's within the monocots. More recently, mycoheterotrophic cheaters arose within dicots, but then we see that it was always linked to a shift to the fungal associate of microheterotrophic monocots. So they repeatedly targeted the first group that was targeted by the oldest microheterotrophs in the monocots, in a kind of convergence in terms of host range. We still don't know what is the feature of the fungi. We still don't know why the plants do not avoid those fungi that likely are more prone to lose carbon, which they give to the microheterotrophic plants. And especially how mycorrhizal networks can still remain on average mutualistic despite they are facing microheterotrophy is not very clear. So we still have a lot of open questions, but you see that since the first land plant interacted with the ancestors of glomeromycota, a lot happened. A lot happened not only in terms of who is associating with who, but also in terms of functioning. And that was the short story. So short stories of evolution I wanted to stress out. I would like not only to thank my collaborators and PhD, but also to thank the International Society for giving me the opportunity of making an update on those few stories. And also, I would like to thank you all for your attention.
Thank you, Marc-André. There is a question for you. Is there evidence of mixotrophy in arbuscular mycorrhizal plants?
So yes, that was what I was showing at the very end. We have, if I understand well, the question is on microheterotrophic plant in arbuscular mycorrhizal symbiosis. And then yes, the answer is yes, definitively. That's the last one I showed. There are some gentianaceae and some polygalaceae, which are microheterotrophic, plus some groups which are quite obscure to those of you who never went to the tropics, like Burmaniaceae or Triuridaceae. And definitively, yes. To my knowledge, every mycorrhizal type was cheated. Every mycorrhizal type has its own microheterotrophs.
Thank you.
Thank you for your help, Silviana.
Well,
Thank you, Mark, for such an illustrative presentation. And I was just watching how you simplified the concepts of origin and evolution of mycorrhizas and focused on the cheating aspect of that. That was commendable, meaning I was just appreciating for the audience, it would have become very simple for them to understand. Now we move on to the next segment, that is the open discussion session. So maybe you can choose your questions which you want to answer live. I'm just requesting all three of the speakers to choose your questions which you wanted to open live. So I would just request Virginia also to put her mic on screen on so that she can also join in this discussion. And maybe you can address to your audience directly, referring that this person has asked this question and I would like to answer it this way. And I would just request, I think that should, maybe I could request Christina to just moderate this open discussion for 15 minutes. Christina. I don't know whether she's here or not.
Yes.
Yeah, she is there probably.
Yes. Yes, please. We have a lot of questions to Professor James White. The audience was very alert and active at the beginning of the section. So maybe you could choose one or two of these questions.
Yes, I don't see any of the questions. I see comments, but I don't see any questions that that address me. You'd have to read a couple.
Okay, so I will choose one. Is rhizophagy common in any terrestrial plant?
All plants that produce root hairs actually will do rhizophagy. At least that's our evidence based on our experiments using plant seedlings. Okay, that means basically that everything from fern allies, where we've seen the microbes in the roots, all the way up to more advanced dicots and monocots. There is a cavet I will use, that I will add, that the function of providing nutrients to plants actually moves to the mycorrhizae in more mature parts of plants, and in certain plants, for example, plants like Ericaceae that grow in acid soils. Acid will actually inhibit the rhizophagy cycle. It will prevent the microbes from entering into the roots, and instead you have fungi that will grow in there and basically replace in its entirety, replace any of these microbial activities. But if you grow Ericaceae on non-acid soils, you do get rhizophagy. At least that's what I'm told by people who've actually done that. So, to some extent, it's environment. To in another way, ectomycorrhizae can totally replace the function, and maybe much more efficiently than rhizophagy cycle. But all plants seem to do it. Any plant that makes root hair seems to be doing this. And while we haven't known about this a long time, our experiments suggest that it's a common phenomenon in plants that we know very little about.
Mm-hmm. But do you have any idea about, for instance, the percentage of nitrogen that the plant can get from these mechanisms?
It really is an interesting question. We're playing with this, trying different plants in different situations. The experiments that we have done so far suggest that it could be 30%. About 30% of the nitrogen that comes from this, but it might depend on the microbe. So we can't really, definitively. We've done some nitrogen 15 transfer experiments where it seems like 90% of or 30% of the nitrogen is transferred. But in other cases, it may be more, it may be less. We've done a few experiments here, and it seems to vary depending on the plant. From each experiment, it seems to vary depending on the plant, depending on the microbe. So we don't have terribly definitive information that we can provide.
And in terms of energy costs, would these be more costly for the plant than nitrogen fixation, than supporting the...
They would be less efficient. Would they be? I think they're cheap. It's comparable to having nitrogen fixing symbiosis in that it costs photosynthate, and oxygen, molecular oxygen is cheap. It's from the air, so they just take that molecular oxygen and make superoxide, and then zap the microbes with superoxide, and so long as there's plenty of oxygen in the air, it's fine. Now, what happens is when carbon dioxide becomes elevated in the soil environment, that will actually inhibit formation of superoxide. And then you don't have this efficient oxidation of microbes happening. So there's some evidence that changing in experiments that people are doing with elevated carbon dioxide, that they see inhibition of nutrient extraction from soils. We think that actually is extraction from these microbes. But all of our experiments are still, many of our experiments are still preliminary. So we're still trying to figure out, exactly what's happening. We do know that we can elevate carbon dioxide and totally inhibit rhizophagy cycle, superoxide formation, rhizophagy cycle, and totally inhibit extraction of microbes, extraction of nutrients from the microbes. And in fact, truncates even development in seedlings or in roots, because you don't have root hairs forming if you remove these microbes, or if you inhibit the rhizophagy cycle. Those microbes are necessary to have proper root development and root hair formation. So it's actually a fundamental process in plants that we're still trying to figure out.
Mm-hmm. So last but not least, Julieta Alvarez is asking if this ejection of microbes is involved with rhizal deposition, or is part of the rhizal deposition.
Say that again, with rhizo, part of
So
rhizo.
if the mechanism of microbial ejection that you showed through
Yes.
the root hairs, if
Yes.
this is part of the rhizo deposition.
It is, I'm fairly sure it's part of populating the rhizosphere with microbes that are adapted to the plant that might function in this rhizophagy cycle. Those same microbes actually will go out into the soil and will colonize, in some cases colonize fungi, for example, cause those fungi to leak nutrients. And then that weakens the pathogenicity. It makes them avirulent because they're leaking, they don't have enough energy really to cause disease. So it changes that whole soil community of microbes and the relationship to the plant. Few pathogens when those microbes are there. Mostly pathogens are converted into non-pathogenic microbes.
Mm-hmm. Okay, I think we have to move. We still have a lot of questions, but we have 15 minutes. Let's have 5 minutes for each of you. Thank you very much. It was fascinating to have all these possibilities that we are not aware, and they are relevant in these cycles. Professor Virginia was, I don't know if you can see your questions.
I answered most of them. There's one, so under the answer link, I've already answered a bunch by typing. There's one new one, and the question is from Maya Rail saying, Dr. Weiss, what are the main specific objections, not just general ethical questions, to genetic engineering or assisted evolution of corals to increase resilience? What concerns do you personally have about this approach? So yeah, I mean, there's a lot to talk about here. I would say, let's take it away from controversy for a minute and talk more generally about solutions, which when I first heard that term, I thought, well, that's a silly euphemism. You know, what does solutions mean? But now I embrace the term, because the term is really referring to a variety of approaches, a huge variety of approaches. It could be, you know, putting up shade cloth across acres and acres of coral reef. It could be seeding reefs. It could be genetic engineering. It could be germplasm repositories, which I'm interested in. These are all in total aimed at helping coral reefs survive. The reality is we're going to lose coral reefs in this century. That, frankly, is a reality. Corals are not going to go extinct, but reefs are going to go away in many, many, many places. And in many places, they're already gone. And so the topic really needs to move away from pristine biodiversity and move towards people. How do we help the people who rely on reefs survive? How do we prevent people who live in nations where there are reef islands? How do we allow them to stay in their country and not have to extirpate their nation because of water rising, you know, these kinds of things. And so there are lots and lots of examples of things that can be done locally that are not scalable on in a larger reef context. And so my emphasis here is, you know, I'm not a field biologist, I'm a cell biologist. And I, you know, sort of woke up and smelled the coffee a couple of years ago and realized, you know, it doesn't matter if I'm a cell biologist, I've got to be helping. We all have to be helping in the ways that our expertise helps us, enables us to contribute to the issue. So then to return to your specific question about, do I have reservations about assisted evolution? I think that I have fewer reservations than many, because this idea of a pristine reef, frankly, we don't have pristine reefs anymore. It's been a long time since we've had pristine forests. Think about, like, in the United States, we have acres and acres of managed forests. And there's, you know, that's the reality that we live in the terrestrial world. And so we have to focus on, yes, we need to preserve biodiversity. That's why I'm interested in germplasm repositories, because we're going to lose a lot of species if we don't do that. But we also have to worry about the fishermen in a developing nation who is relying on protein from the reef that has gone away. You know, what are the mechanisms that can be used to help him or her survive and feed his family? And so it's in these small scaled contexts where things like assisted evolution, where a set or a small population of corals could be useful in helping a reef that's not pristine, but a reef that exists, period. So again, lots and lots one could talk about, but you know, there are many solutions that we all should be working on.
Mm-hmm. Mm-hmm. Okay, thank you very much. So we have here a question about microplastics and how do they affect the coral reef or the symbiosis. Do you have any?
You know, that's a great question. There are a couple of them in there about microplastics. You know, it can't be good because these are, or you know, the cnidarians are they're predators that bring in food by, you know, engulfing and phagocytizing food. And so surely they're phagocytizing microplastics. I honestly don't know much about that. I don't know of folks who were studying that. I think with corals, you know, there's this giant gorilla in the room that everybody is paying attention to, which is global warming. And all of these other insults, which are also very important, like, you know, ocean acidification and microplastics and all these other local effects are also important too. But, you know, there are few of us. There are probably people working on that. I don't know of them.
Mm-hmm. Okay, thank you very much. And it is a fascinating world of symbiosis, very pretty and very exciting. So thank you very much. And then for Professor Marc-André Sellos, that showed us in such a clear way the symbiosis and the mycorrhiza, and then mixed it all at the end. It was very interesting and very new for me, at least, way of viewing the world of mycorrhiza. And I would like to ask you if you see these mycorrhiza as a network of carbon that is going from one plant to another, and also from fungi to fungi. Do you see that this carbon is a cost for the plants, or it is the carbon that is circulating and the nitrogen, the nutrients that are circulating, are they just a surplus from ones that can be used by the others? What is the cost, you know, these market in the symbiosis? Do you see it, or do you see as just this system using the carbon that is a surplus from the photosynthetic fixation by plants?
It's a very interesting question and a very relevant one, but I'm afraid we cannot answer that very properly so far. We are missing key data here. The first one we are missing is what is the impact on the fungus, which is, I said, cheated, but we can guess that if the fungus is giving carbon, it's not very good for his physiology. But do we know it? No, we don't have data. We don't have impact of microheterotrophs on the fitness of the fungus. More concerningly, we don't have impact on the fitness of surrounding plants. So a postdoc that did not finish his work in my team was doing some experiment in the forest, and we had the feeling that microheterotrophy is quite negative for successional species, but much less for the species at the climax, when the vegetation has been reaching a climatic stage. That was very preliminary, and I would not be very sure of that, but really it's hard to investigate, but we don't know. if there is a price for the surrounding plants. So now, shifting to the question of what's moving in which direction. There is not only carbon, but also it seems sometimes in some experiments, nitrogen or even phosphorus.
Phosphorus? No.
They're moving, yeah. Yeah, Sinua He was publishing a couple of nice papers on nitrogen in phytology some 15 years ago, I'm afraid. It's not very recent. So we have evidence for that, but it's rather experimental evidence and it's punctual measurement with labeling. And the most important is to have a budget. That is to know over the life of the forest, over the life of an individual, over a year, what is the net flow. For microbial networks, it's very interesting to have all those papers with labeling, okay? Because we know that it means carbon or nitrogen can move, but we need budget. And let me tell you that we have a budget when we have a plant which has an enrichment in carbon 13, because of that we can say, okay, this percentage of the biomass is from the fungus, while this percentage is from the... But if you put aside this very extreme phenotype of microheterotrophic and mixotrophic, for normal plants, which are coexisting, two trees, one next to another, we have evidence of transfer, but we have no carbon budget. We don't know if sometime one is paying for the other. Actually, in Switzerland, a climate change experiment showed that on average, trees are giving a lot to the neighbors, but they receive nearly the same. So, except some specialists of using the network as a carbon source, it's still not sure that everyone connected to this network can have an exchange.
Okay, thank you very much. And it was a pleasure to be here, and thanks to all that took place and organized it. Thank you.
Merci.
Merci.
Yeah, thank you very much, the speakers and Christina for taking it so nicely with all the three speakers and covering all the topics. And I hope everybody took maximum benefit out of this discussion. Now, having listened to our experts and speakers, now this is time for students. And I'm glad to welcome Miss Ashley, a student representative in ISS, to share her opinion or any talk which she wants to address to the student members of ISS. Ashley.
Thank you, Manju. I'm just going to share my screen. All right. Good morning from Melbourne, Australia, everyone. My name is Ashley Dungan. I am finishing up my PhD, just a couple weeks away from submitting my thesis, which I think puts me in this really interesting position where I can sort of understand the things that have been beneficial to me thus far in my career, but also looking forward, I certainly have an understanding of what I feel are sort of skills or aspects that I might be missing when it comes to moving on and looking for a postdoc or moving into employment. So sort of in that context, we've been thinking about what ISS can provide to students. And I've really broadly characterized the things that students or early career researchers or ECRs might need into these sort of three broad categories represented by these interconnected circles. And those would be mentorship. So the ability to connect with and learn from more senior scientists, something that we're certainly sort of missing a lot in the year where we can't get to conferences. Skills that relate to employability. So those might be training workshops, the ability to give a presentation to the scientific community, those sorts of things. And really importantly, enjoyment. There's a lot of things that ISS might be able to do to help remind us sort of why we got into symbiosis, why we've created this really long-term commitment in our careers to begin with. So what I want to do is go over a couple of ideas that we sort of put together for events that ISS will be able to, in a virtual way, host in the short term before the end of the year. And at the end of that, we'll put up a poll, and I'll ask that all the participants sort of vote on which option would be most beneficial to either yourself as a student or early career researcher, or to your students or other mentors that you might have. So the first would be a photo contest, similar to what was done in the 2018 ISS contest. We would like to open up a photo contest with the prize being free registration to the 2021 conference. As well, winning photos would be used in advertising materials and proudly displayed during the conference, with the possibility of being included in the proceedings that are written up after the conference in Lyon. The second being virtual coffee roulette. So this is something that I've participated in here in Australia, and I found it really quite fascinating, where we were able to, as students or early career researchers, be matched up in a casual way with more senior scientists, and just to have pre-arranged Zoom meetings where we could just talk with other people in our field, or maybe similar, we all share that symbiosis theme, but to talk about something in a more casual way, get to know people in the fields, and perhaps make connections that you wouldn't otherwise be able to do. The third being a writing workshop. So this would be a little bit more of an intimate arrangement, but still held virtually. I can imagine hosting a couple, since we are this internationally distributed group. But here, students or early career researchers would be paired up one-on-one with a more senior scientist, who would be sort of reviewing parts of a manuscript or maybe a thesis chapter that they're working on in real time. And so within a week, you would have someone that is leading you through this writing process, but also reviewing your work in a really supportive way with, zero judgment. And the last would be similar to what Manju has magnificently orchestrated here with this webinar, to have a student and ECR-led webinar. That means that not only would students be doing the organizing and selection of speakers, students would be doing the speaking. And those might be long 15-minute talks, or just short 3-minute talks with sort of cash prizes, if you will, for the best speakers of different categories. So Manju, if you would,
I can launch your poll.
Okay,
Yeah,
that'd be great.
It is going. Yeah, let's see.
So if everyone wants to select one of those and submit, that would be really helpful for me. So the last thing I want to mention is, my role in this committee. I'll say thus far, mostly it's been things like social media, but to create any of these events, we'll certainly need participations from the international community. And so I'd really encourage anyone who wants to get involved, who wants to meet some of these people who are on this executive committee. If you look up to them and you're really excited about maybe talking with them, try to get involved. It's not just good for your CV. It definitely is beneficial there, but it does provide. You do get a lot of mentorship and support. I don't think that I'm alone in thinking that a couple of years ago, I would have been absolutely fangirling over the chance to talk to someone like Virginia Weiss or Simon Davey, because I do work in corals. And now they know me by name, and that, as a student looking forward and trying to find a postdoc, that's something that's really exciting. And I really want others to be able to sort of make those connections. And so in the bottom left-hand corner, I do have my email address, and please spam me. I would love to do nothing else than answer your emails, anything to get away from writing my thesis today. So please contact me, reach out. We'd love to have you as part of the committee, part of finding ways to get students and ECRs to be able to connect with one another, and really sort of advance our professional and personal success in all symbiosis research. And that is it.
Yeah. So I end this polling.
Yeah,
And
I did.
I can share the results. The results of this poll. I think everyone can view the result of this poll is
All right.
that students are very much interested in the webinars.
Yeah, but you know, honestly, there's a lot of pretty evenly distributed otherwise down in the other options as well. So
Yeah,
that's really
yeah.
great. All right. So I
So-
have all your email addresses. So look forward to hearing from me in the near future. Great. Thank you,
Yeah.
everyone.
Yeah.
Just,
So time for the next conference. I would just invite Professor Mark Andre to share his plans about Lyon conference in July 2021. Yeah, please.
Okay, yes, that's okay. I'm looking for the file.
Yeah.
I'm just a bit lost with the windows on my computer. So, as you know, we will have next year a symbiosis meeting, but it will be a quite special one because this year we will have also a joint meeting with the holobiont community that has its third meeting. It's very important because, you know, holobiont, it's a matter of symbiosis, and our communities are evolving in parallel. They don't cross an house. So we decided to, it's coming soon, share the, voilà, ça y est, finally, you have it. To share the 10th, it's the 10th ISS Congress with the holobiont 3 Congress. And it will be in Lyon. Lyon is central France, close to the Alps. It's a wonderful city, a bit of touch of Mediterranean atmosphere, a high place for gastronomy, but also a high place for culture. On the bottom right, you have the Musée des Confluences, a huge museum of natural and history and human sciences. So it's also a place for tourism. That's what I mean, if you want to stay 1 week after the Congress. We will meet at a very big meeting hall where we will have huge space, not only for scientific events, but also for a bit of outreach. We will pay attention to that. So this meeting is, well, that's a slide which is not very pedagogical. By the way, we'll have a lot of sessions. And this year we call it Symbiosis in the Anthropocene, because exactly as Virginia was nicely showing, there are emergencies facing which our knowledge is useful, but should be immediately and quite quickly implemented. So Symbiosis in the Anthropocene will have some quite up-to-date focus which are socially relevant, such as One Health and the holobiont session. Another one on agricultural relevance of the symbiosis with plant holobiont, physiology, defense, and applications for agriculture. Well, if you want to know more and see exactly what are the sessions and also what are the invited speakers, the website is launched. So I'm pleased to invite you to have a look at this website. We are trying to prepare something scientifically relevant, socially relevant with fun. So we hope to have you there. We do hope to have you there. You're more than welcome to join us in Lyon. That's it. Merci. To see you soon in Lyon.
What is ICR? What is ICRC? Yeah. So
ICRS, the International Core Reef Symposium, was meant to occur
Okay.
right now, like
okay.
yeah. And
Yeah,
they
we
placed
have an overlap.
They placed it immediately. They placed it exactly on top of ISS.
Okay. Same.
Yeah, but
And
it's because-
Simon tried, but
It didn't
Yeah.
work.
there's nothing we
Try
can
our
do.
best. The fact is that it was postponed because it was due for this year, but with the COVID issue, it was postponed, and unluckily, it's the same week. So we are
Yeah.
trying now to organize a kind of dual inscription registration for people to be able to have remote contact for some sessions. We'll do our best, but it's not optimal.
So what has happened is the most important member who deals with the money and everything of ISS is, is his turn is to come because he is going to look after everything. So what I do now is I will go according to, thank you, Mark, for briefing everyone. I hope everybody will be quite interested in our conference and they would join you at Lyon. Now, before I invite Eric, I have one more thing in the line is that I got to invite Professor David Richardson, Editor-in-Chief of journal Symbiosis, to share his words of wisdom and recent developments in journal Symbiosis. So, David.
Thank you, Manju. Can you turn my video on again? You've turned it off.
I can do that. Yes, please.
Yeah.
What is that? Yeah.
Okay.
Yeah, here you go.
Hello, everybody. My name is David Richardson. I don't know whether you can see me because my screen has changed,
Yeah,
so I can't see you.
sorry.
But that is okay. Can you see me?
Just try, it will work. Please try putting your camera on.
Okay.
Yeah.
Anyway, my name is David Richardson.
I think you could try. Give it a try.
Okay, wait a minute. What do I press here?
Otherwise, I would do it from here.
Oh, wait a minute. Ah,
yeah,
right.
There it is. Yes. Yeah.
Okay. Yes, the journal Symbiosis focuses on the interaction of the partners in a wide range of associations that involve animals, plants, and microbes, as you've heard this afternoon. We're associated with the International Symbiosis Society and currently published by Springer Nature, which is one of the leading journals of the world. Oops. And now, okay. So Symbiosis, we have 9 issues a year, usually between 8 and 10 papers in an issue. And these 9 issues are separated into 3 volumes. And the regular issues of Symbiosis cover the range of research papers, as you can see here, reviews, research papers, short communications, and book reviews. But we also have special issues. Every 3 years, we have publish the proceedings of the International Symbiosis Society congresses. And we also have special topics. Oops. In the special topics, we've got one forthcoming one on lichens. We've published several proceedings of the African Association of the Biological Nitrogen Fixation. The Spanish Society for Nitrogen Fixation as well. But we do have particular issues devoted to particular symbiotic systems, as you can see here: nematode, bacteria, symbiosis, exploiting mycorrhizal associations, for which you heard a little bit this afternoon from Marc-Andre, and Frankian actororhizal plants. I'm the editor-in-chief, and here you can see me making a toast to the World Symbiosis Day. The reviews for the Symbiosis are handled by Dr. James White, who gave us a talk today. We try to have at least 1 review per volume of the journal, so that's at least 3 issues, sometimes more and sometimes twice as much in each year. The special topics the keywords in some of these reviews include as common items, the list that you see here, from endophytes to Wolbachia, from coral systems to lichens. If you submit a paper to Symbiosis, then research communications and short communications are reviewed and revised with the help of the editorial board members, acting as manuscript editors, and they take the papers through the review process. And we have 29 members of our editorial board, and they come from 15 countries. So we have a great diversity in countries and a diversity in the subject matter of the symbiosis that they are expert in. I should tell you a little bit about how symbiosis started. You had one or two words earlier, but in essence, Professor Margaret Gallun, as you see here, seated here, was doing research in the early 1960s on lichens and other symbiotic systems. And she decided to review what was in the literature, and she found that the papers on symbiosis were scattered through a whole wide range of journals and difficult to track down. So she said, well, why don't we start a special journal where all the people who are interested in symbiosis could send their papers? And she started the journal in 1985, published by a colleague of hers, Miriam Balaban, of the Balaban Publishers in Israel. And so that worked very well, but then authors of papers, submitted papers, said, well, you know, it's all very well to submit our papers, but we'd like to get together and talk. And so she organized in 1991 the first International Symbiosis Congress, which was held in Jerusalem in Israel. And actually, the symbiologists from all around the world flocked to that meeting, and the proceedings for that meeting amounted to 500 pages. And then the second International Symbiosis Congress was organized in 1996. And people said, well, it's all very well that we have these congresses periodically, but wouldn't it be nice to have a way in which we could interact on a daily or more frequent basis? So it was decided by the participants at the second Congress, and I was at both of those, in 1996 to start an International Symbiosis Society. And you can see the picture here of Dr. John Lee, who was the first president. And as you've heard, the congresses have been held every 3 years since then, and we're coming up to the 10th Congress. The journal Symbiosis, if you join the International Symbiosis Society, which I hope as many of you who are listening can do, then you do get access to the current issues published by Springer free as members of the ISS. In addition, the contents from 2009 to the present can be searched, and I give you the search site here, by those who are members of the ISS or by others. But if you're a member of the ISS, you can download and see the papers. Now, the issues from 1985 to 2008 were published in hard copy by Balaban Publishers, as I've mentioned, but they now have all been scanned and they're online and they can be searched for free. So you can search even the proceedings of that first Congress and all the others just by going to this next website. Well, what about the future? We continue to plan to publish the regular issues, the special issues, and we hope to have a special issue of Symbiosis in honor of the World Symbiosis Day. This will provide an opportunity for mini reviews by the speakers today, if they wish. And also many of the questions or thoughts expressed by those setting questions to our speakers today might be included in that special issue. If you wonder what Symbiosis looks like, here's a picture of 3 recent covers. As you can see, we cover a very wide range. On the left-hand side here, you can see a picture of lichens. And what happens is that when each issue is compiled, I look through the papers that have been submitted, and select a photograph of a key paper for the front page of the journal. And you see on the left one for lichens. In the middle, there's one which involves a tree with nodules on its roots and nitrogen fixing. And then on the right-hand side, you see bark beetles and their associated fungus in the symbiotic system that they're involved in. So that we have a nice green cover and a nice pretty picture for every issue that we publish. If you want to find more about the journal, there's a couple of references here on the history of the journal. An obituary for Professor Margaret Gulun, a wonderful lady with great foresight, and then the journal, the paper in which I and others from the last Congress published, where the proceedings were published, an introduction thereto. So that's the end of my presentation, and thank you for listening.
Great. Manju, can I just go?
Yeah, just hang on for 1 minute. Just wait. Okay. So, David, can you remove this slideshow? Can you just stop this?
Yeah, I'm just trying. Stop. That's it.
Yeah, thank you. Thank you, David, for such an exhaustive expression of the journal Symbiosis. Now, before we conclude our before we conclude our grand knowledge enriched event, I would like to invite, and he's there, Professor Dr. Eric Home. He is the treasurer of ISS, and we have done good work in his presence. So we have, yeah, he would like to show some of the part of ISS with the help of maybe some PowerPoints he has. And then we have a question answer poll. I would love to take feedback from the participants. So just wait for that. And then I would cast the vote of thanks to all of you. So I would give it to Eric now. So let us see what he wants to say about ISS. Eric.
Thank you. I'll keep this short. But if you've enjoyed our time together and in interacting with some of us, I have, I invite you, if you are not already a member, to join our society. As was said early on, we do value diversity, especially geographic diversity, and makes symbiosis fun and makes doing science fun. So here we have our website, and I'm going to put the links into the chat for everyone in case you don't have it. So I just did that. And so the website, on the website, I just want to highlight a couple things. You can go peruse at your own time. But here's the website. You can access the Symbiosis Journal, as David was talking about. And you should know that our publisher, Springer, will actually give a book prize every year to what they would deem the best paper in Symbiosis. And they will also give a book prize for our posters in our meeting, the ISS meeting that Marc-Andre told you about. On the main page, we have our Twitter feed. You can subscribe. But I want to talk to you about membership, and it's this tab. You just press member. We have different membership levels. If you are from what we call a global outreach country, any of these countries here, we have lowered the membership rates for you. And those options are shown here. You have a normal 1-year membership. If you're a student, there's also a rate, whether you're a global membership or a normal student membership. All the way up to, you could have a recurring membership up to 3 years. So we encourage you and invite you to be a part of that. And lastly, I'll just highlight, we have a listserv that we would like to use and have more dialogue with. And you can sign up on the link that I also mentioned there. And that just goes to a group of people within the society, part of the society, in matters of symbiosis. And that is it. I think I need help.
So, but I need your help. So do not leave the screen because I need you for discussion about these questions, and I also need you for the concluding the seminar, right? So you just be there. Maybe if you want to stop the slideshow, that you can do.
I will do that.
Yeah.
Yep.
So do I launch the poll, Eric? We had decided three questions.
So we will part with just a few questions. It's not very long, but it would help us very much in helping you and helping the society.
Please participate in the poll. That will genuinely help us in improving ourselves. And I'm allowing all the panelists also to vote. So please, everyone participate in this launch poll, which I am launching. So we have three questions here, and please.
As people finish up, I forgot I should have emphasized, your membership gives you access to all past and current issues digitally of the Symbiosis Journal. So I want to make sure I said that.
And Eric, you can take on the poll result also, how they are feeling, and according to the response. I'll just launch the results after this is done. 71% has voted. I still hope for a few more people to join for voting. A lot of people have left already. So there are two things remaining now. One is I want to have a group photograph or maybe a screenshot at the end when all of us are present together. And then I want to thank everyone individually. So after this poll is over, I think it is almost done because it's not increasing. I would just end this and share the result with everyone.
You've got my...
Yeah, Eric can explain the result and express his views about that. Eric, the poll results? Your mic is off.
Well, it looks like, thankfully, most of you would like to join or renew your membership. If you're undecided, we're happy to talk with you about it. And hopefully, we'll see at least a quarter of you at the meeting in Lyon. At this stage, you don't know. We may not know also, but hopefully there's actually going to be some remote option given the new realities of COVID-19. And it looks like most people would like to see more research papers in symbiosis.
Yeah. So thank you very much, everyone, for taking out time to answer to our poll. This is really going to help us improve ourselves maybe next time. So when Chris is also around, and I just thought at the end, I want to say thanks in my own way. So I have, first of all, I would like to express my gratitude to the guest speakers today who condensed all vast world of symbiosis in 15 minutes at my request. But it was very enthusiastic for the audience, and I could appreciate that the time was short, but still we could cover many aspects. But I'm very thankful to you for having agreed and for condensing the information to a shorter time. Then I would like to thank Simon and Eric for being especially supportive to bring shape to this event. Otherwise, this would not have been done like this. Then to David Richardson, probably his camera is not on.
No, you've switched my camera off.
Now you can come, please. Some control is there with me and I'm going to enjoy that. Yeah. So David, I want to thank you that you coordinated everything. He got a lot of response from a lot of countries. And also he arranged everything with Springer Journal so that Springer Nature, so that it comes out to be smooth. Then I want to thank Silvana and Christina for having agreed at the last moment as well to arrange and organize. And it was a very good get together for ISS executives. Also, we had not seen each other for quite some time now. And then I convey my special thank to Sharon, who got all website and everything renewed at this time of work so that, we could get things done. I also take this opportunity to thank Professor Christopher Walker, who made this event special for me for being present here. Thank you, Chris. Then I have few students, and my colleagues at Sri Aurobindo College who helped me to come up with this event. And all through, Ashley was there with me to conduct all the activities and this email business and everything she was doing. But a lot of work, other work was also there, which was being done by some of my students. I'll just check if they are there, so they could also join us in the last moment snapshot. And I just want to summarize whatever we did for our audience. A lot of them are still there. Maybe that I can do at the end. Maybe. So I would just check whether they are there for this snapshot, last snapshot. So they should be here.
I can't have my video on.
Okay, Christina, just hang on for 1 minute. What is his name? Yeah. Then I have. Yeah. These children, they really worked with me all through and made this success because they've been working with all. One was specialist in advertising, the other one was in analyzing the Excel file, and the third one was taking care of. And they have all decided that they'll be preparing a very good report after this all is done. Right? So maybe Sarthak, you can click the snapshot from your, and then later maybe we will be able to distribute and take it a good one. If I'm missing someone, just hang on. I'm missing who? Christina. Yeah. Sorry, Christina. Sorry.
No problem.
And then Hema also I'm missing.
Hello.
Yeah, I think she is not doing it.
Ma'am, I'm here.
Yeah, the video was off. Yeah.
Ma'am,
so
you stopped my video. You stopped my video.
What has happened? Yeah, it has come.
Yeah, it's on now.
It's there. So Sarthak is going to click a good snapshot for all of us so that we can print this in our report that these were the panelists and the support group for this webinar. So I think he's done. Are you done?
The count of 1, 2, and 3. Go.
Yeah. Thank you, everyone. And thank you,
Thank you,
everyone. once
Manju.
again.
Manju, could I just say thank you to Manju and her
Yes,
team.
here, here.
Because
Yes.
Manju actually is the one that came up with this idea and really did all the hard work with her students. It's been a great idea, and I think there's a lot of things to take away from this. I think we're going to try and do this each year. Obviously, we have to work it around the Congress as well. But really, obviously we had a lot of introductory material in this one, introducing people to the society. But I think moving forward, the research talks, what a fantastic idea, and the discussion that those generated, and I think freeing up time for more of that and in the future, now that people have got to know the society moving forward. And I want to thank everyone for joining in and participating. So with that, but thank you, Manju. Thank you.
Most welcome, Simon. For any
Thanks a
work,
lot.
I am always there. So I hope to see everyone again in Lyon conference. And I have a small, PPT which my students have prepared how to end this program with the audiences. So I would just run my PPT and say bye to everyone. Thank you once again. Thank you very much.
Thank you.
So
Bye-bye.
I would just
Bye.
share screen. Bye, everyone. Bye. And play that PPT. Yeah. Thank you, everyone, for joining us today. Here are some ways to stay connected. Become a member of ISS. Publish with us in journal Symbiosis. Join us in our 10th conference at Lyon, France in July 2021. Thank you, everyone being present here, and especially to the overwhelming response from the scientists in different countries. That made our effort successful, and we look forward to see you again. Thank you very much, everyone.
Bye.
Thank you. Bye.
Bye.
Bye-bye.
Bye, everyone.
Bye. Thank
Bye.
you.
Bye. Thank you.